And while we're waiting, since this is being recorded, we'll just do a little, as they call it, a filler. What's beautiful about this particular workshop, we get a chance to see NOFA's work in action in the city of Springfield. So, and it's really an honor for me to get a chance to work with my dear friend and brother, Andrew, to work with this wonderful project and to engage with Agnes and the other youth leaders at Home City Housing and GTC. They're doing a bang-up job and really have taken and made this project their own. So I'm looking forward to the discussion today and having our consultant, Nancy Clem, who I have learned a boatload of stuff from, and has done a marvelous job working with us from afar, coming out of the Chicagoland area. So it's great just to be on here with everyone, and definitely Christine, who stepped into the use of Carol Roselle, taking over all the and just knocking it out of the ballpark. So this is really what I call the power hour with the A team in the house. And so it's definitely a blessing to be here with everyone. And, you know, technology does what it does, and we're going to work through it and have a good time with it. So those who see this recording will see the show does not stop. We keep it going. And we are going to definitely learn a lot from today's, really from today's presentation that will help all communities, but particularly in our cities like Boston and Baltimore and Springfield. And it's definitely a blessing to talk, to have this conversation. I think it's much needed and right on time. Welcome, welcome. You have joined the Bioremediation Workshop, our second year Bioremediation Project in the city of Springfield, Massachusetts. I'll introduce myself. My name is Anna Gilbert Muhammad. I am the Equity Director and Food Access Coordinator for NOFA Mass. And I am, as you can tell, I am based here in Springfield, Massachusetts, which is Pecumtuck land. And so happy to be here today and happy that you have joined us. You are in for a wonderful treat. And today we'll be talking about the second year of the Bioremediation Project here in Springfield, Massachusetts, one that is really youth-centered and youth-led, led by our very intrepid Bioremediation Coordinator, Andrew Laurian, who serves as the Bioremediation Coordinator for NOFA Mass. He is also the youth manager for Gardening the Community, so he's wearing two hats. And we're glad to have him do this presentation. We're also joined by our consultant from this project, Nancy Clem. Nancy is actually out of the Midwest, and her work lies at the complex intersections of the theory of practice, and practice of land politics and soil health. Her work as a communicator, consultant, curator, translator, provocateur, and medium is intentionally recognized in communities of practice, ranging from publishing to fine art, justice activism, to philosophy, growing, and to podcasting. She splits her time between Little Village, which is a densely packed, diverse urban neighborhood in the heart of Chicago, and with Driftless Region, which is 50 acres of land in Northwest Illinois. So she is from the Chicagoland area. And on that particular piece of land, she stewards a prairie, cultivates and forages medicinal and edible plants and fungi, keeps bees in the fruit orchard, raises quail, and grows for a seed bank. And so her work really is at the intersection of urban and rural lives, and she takes a very holistic approach to soil health. So we're happy to have her with us today. And so as we are working to get our slides up so you can see some of our sponsors as well as our land acknowledgement. Now we are broadcasting today. I am broadcasting from what they called at one point Pecumtuck land. It is still Pecumtuck land. And we will put up a map shortly to show you where you might be located, what indigenous family are in the area that you are currently living in. And these were lands that were inhabited and managed by our First Nation peoples before European colonization. These families are still here. and they're still working, but have faced a great deal from white supremacy, racism, death, disease, but they are still resilient in their work. And what we're asking as NOFA Mass continues in their growth in terms of dealing with racial equity, particularly racial equity in the food system and in agriculture. We acknowledge that after years of racism and enslaved persons suffering, that there are corrections that have to be made, and it is definitely NOFA Mass's intent to continue that work both internally and externally. What we ask is that you join us in a call to action to support these organizations, to provide whatever resources you can, whether it's money, whether it's land, whether it's tools, whether it's your knowledge, because there's definitely a change that has to be made, and NOFA is trying to help in that change and be a partner with these different organizations, such as Gardening in the Community in Springfield, the Urban Farming Institute in Boston, the Native American Legal Fund, and a host of other organizations, BIPOC organizations. So without any further ado, lastly, we'd like to thank all of our wonderful sponsors that have contributed and continue to contribute to NOFA Mass. They have been generous supporters of our conferences, of our organization. Chelsea Green, who has always been there, the Food Co-op organization, Johnny Seeds, and so forth. Those different sponsors are also attending as vendors. So if you want to check out the vendor showcase, please support our vendors. And then lastly, please support the auction. They have some great events. We'll put those links up in the chat so you can see it both at the beginning and the end. But without any further delay, I want to turn everything over to our presenter today, Andrew Laurian, who will take us further. Andrew, I will make you a co-host and you can go right ahead. Okay, so it looks like Andrew is back. He is just connecting to audio. And while we wait, we're also joined by one of our youth leaders. We call him Youth Agricultural Scientists at Home City Housing, which is the partner organization for this grant. This is a Massachusetts Department of Agricultural Resources grant that has sponsored this project. Agnes has joined us. She is our lead youth agricultural scientist. She works very closely with Andrew and I on this project. And so if she doesn't mind, while we're waiting for Andrew to get situated, kind of starting us off, giving some of her thoughts about the project, what she's seen, and maybe it'll be nice to get her thoughts on what she's seen so far, since I think she's in her second year with this project. So Agnes, if you want to take yourself off of mute, we can go ahead and start with you while Andrew gets situated.
Hello, I'm Agnes. So it is my second year with the project, and I find it very interesting and going back each week to see the new growth and updates on the project, such as we see a lot of different types of mushrooms that's grown, different wildlife that's now come into the place that was not there before, and to see how the people react to the project happening in their area. We've seen a lot more people hanging out outside near the area, so that's interesting. We've seen bunnies and bees, and in that area there was not any before at all. The plants around the area too have seemed to be affected. It seems a lot better, and overall it's really interesting comparing it to how it looked at this time last year versus how it looked this time now this year, with new growth and how everyone's reacting to it too.
Okay, wonderful, wonderful. I think Andrew is ready. Andrew, you can share your screen at any time and take yourself off of mute and go ahead and get started. We've heard some opening words from Agnes, who is also NOFA's intern. She works directly with me in the food access program. So you get her, it's great to hear her perspective. So Andrew, I will now turn it over to you.
Hey there, can you hear me now?
We can hear you loud and clear.
All right, sorry about that. Wow. So, bioremediation, thank you, Agnes. As you see, you see the importance of this being a youth-led project. This is our future, and this is what it's all about. From what I understand, a lot of the bioremediation work that's out there, if you're doing some research or you're YouTubing, there's not a ton of it out there. So what we're learning, from what I understand, is pretty cutting edge. We're taking basic experiments and making the best of it. So I want to introduce my partners. Our partners in this grant is Guardian Community, the food justice organization engaged in youth development, urban agriculture, and sustainable living to build healthy and equitable communities. Home City Housing are doing great things with the youth programs. Agnes has been a part of that, and Bernard and a few other of our youth that you'll be seeing in our presentation. So to get down to the nitty gritty, currently the prescribed method for dealing with urban soils in Massachusetts, especially those where there is cause for concern of contamination, is to skip soil testing altogether and install barriers, sometimes a thick layer of clay, sometimes gravel or plastic landscape fabric, sometimes multiple layers of various materials, and over the soil and add 12 to 24 inches of trucked-in loam on top of the barrier. The result is a troublesome growing media that can easily become hydrophobic, as trucked-in loams often have high proportions of partially decomposed woody materials and grows thinner over time as the organic components break down or are eroded, resulting necessarily repeated applications of fresh manufactured loam. Furthermore, this approach simply buries the problem of the contamination and delays the need to address it until the time in the future when the barrier inevitably breaks down. So if bioremediation using microbiology and plant partners is effective, such an approach can be beneficial in many ways. And the first thing I can think of is the cost, and the waste. So instead of chucking this stuff out, which is going to cost how much to hire a guy to do that, and then buying the loam or the compost to top fill it, and then where does that stuff end up? In a landfill anywhere, somewhere. So let's be responsible for what it is we have going on. This is our site. As it looks currently, we have a lot of vegetation going on. This is year 2. We have a compost that's breaking down, and we're right here in the heart of the city. This is a very big space that we're able to use, but as you know, it's hard to come by space in the city, which is why it's very important to highlight the community and the interactions we need to have with our rural neighbors. We need to learn things like how to, what inoculants to use and how to deal with certain deficiencies. But first, is our soil, is it even safe to use? We want arable land. We want to think about sustainability. So, what's the future with this lot? We might put a lot of hard work into this lot and then, another house is built on top of it. So, that has a play into food security and having a relevant education to deal with these matters. Community. That's I can't keep it as simple as that, man. If we have that bond between say my rural farmer and I, and he can give us some knowledge, maybe some resources, then also we're a community that needs good food. So until we're able to grow our own, be able to sell our products. So I think neighboring up is the way to go in this project. This is an example of some of the things we pull from our sites. A little bit of this, just a little rubble was pulled from our actual site that we're doing our test on, but this is what it looks like when we're dealing with city soils a lot of the times. We believe this work will stretch beyond the 3 years allotted in this grant. In fact, this work will last a lifetime for some farmers in the city. When it comes down to the spaces we can use, it's 9 times out of 10, we're going to have to do some remediation. And that comes down to, we got to be sustainable. So we have to build. And who's gonna do the work? Our youth. You know, we gotta show them the ropes, let them know how important it is for us to get to where we need to be. And you know, as a grassroots organization, and somebody who's not gonna like invest a lot into these process to remediate the soil, we have to use what we have. So as you can see, we're breaking down pallets. We're learning about safety, teamwork, repurposing material that could become waste. This is one of our first compost bins we built. So we want to build up our compost instead of buying it, $45 a yard on average. From there, we were able to upgrade. We found a pallet that came from a contractor that does siding. So we got a nice long one that we're going to use as the back of a bin system. And in the system, we'll be able to rotate and flip our piles. Hopefully by the end of the flipping, we'll have a finished product. Everybody's really excited. The remediation process, you got to think about is there's a lot of science involved, but also there's a lot more to it. You know, there's building, there's community, there's pride, there's understanding what it is to grow a compost in the soil. Who's living in our soil? Who's living in our compost? Why is it important? Why are we harping on the compost? Well, we're talking about the food system as well. How can we talk about bioremediation and taking better care of our communities than to, like, we have to think about the food system, right? Where does our farm come from? Where does our supplies come from? What processes does it go through? How many transportation units does it have to get through? To continue to build our soil with organic matter as part of the remediation project. That can cost a fortune. So we're all diving deep into the waste biz. Here's our fearless leader, Toussaint from GTC, and we're talking about ways that, you know, waste, we can turn waste into a business. You know, where are we going to get our material from if we would like to compost properly? You know, what kind of relationships can we build? How much can we keep out of the landfill? Because as you know, the waste in the landfill is a big part of our food system. We can't ignore it. We're going through it. We're just giving the youth different activities. Like this group has a business called Just Peachy, where they're selling peaches, and you know, they have to think about what do they need from their farm supplier. How are they going to get their product to the producer? To the processor. You know, what materials do they need to buy to process their food? Who their consumer be? Who the retailers, consumers, distributors? What relationships are you going to build as a business? And then just try to compare that to if we were to buy that same peach in our local store. You know, another group went with Lunar Lavender, and they're making soap products, they're making bath products, and they're really understanding the system and what it takes to and you know, the power of the dollar. And that's what's really important here. In remediation, you can't begin to remediate your soils if you don't understand the power of the dollar. How many dollars do we have to work with sometimes? Yeah, so there's different ways to compost, and we saw this method where we can use a tube that we had found to kind of get air in the center of it. We drilled holes down the inside of this tube, and we're hoping to get good aeration and break down our materials quicker. This is food scraps from our farms supplies when we have an abundance. But sometimes we gotta make it neat. So the youth of our Home City Housing made one that's a little bit different style because we're, you know, in the neighborhood. We don't want everybody thinking that we're just creating a dump site, but we want them to understand compost and its importance. And it could look good too. Now that we understand the social justice that's involved and how important it is to be leaders in our community, we also are meeting our neighbors underground. So underground, we're meeting our soil microbes, and we're trapping IMOs, okay, indigenous microorganisms. And we just set some rice in the ground about 4 to 6 inches, and we buried that, and about a week later we uncovered it to see who was living in it. come up with different colors like hot pink and curry chicken yellow. And it was very cool to just introduce ourselves to the world under. And as the world above, the plants that we are installing into our sites are bringing in pollinators, so it's becoming very diverse. And this is our project plan. That's a rough draft of our plants from our project partner, Nancy. And we have deliberately picked certain plants as a part of our remediation project. Hey, Nancy, are you there? Give us a little chat about why we would pick the plants we picked.
Sure. We were looking at plants that the youth could use as herbal teas that would attract pollinators, and plants that occupy different zones above ground as well as below ground with their roots. And a lot of those plants that we're using are already known as phytoremediators.
That's awesome. And we dug right in, man. Some of it, when you get your hands in the soil, it's a good feeling. So we're here installing plants, some of which we picked of our own. We dedicated one section of our plot to some annuals that we can get in and study. We wanted to plant some things like mustard greens and collard greens that we as urban farmers tend to grow a lot. And we just want to make sure we can understand what our plants are uptaking. So in the future, we'll be doing some tissue tests to determine what it is that our plants are taking up from the soil. We're doing some documenting of everything we see on the site. Nancy made a really good point that has always stuck with me since the first time I heard it, is to approach it and not just look at the problem, but to look at the healthy function. See what's happening right. What plants are thriving in this environment. What is going on that we can see and study from. Here's Bernard. He's been kicking butt over there. He's been with us since the beginning. Like I said, these are leaders. When we're having exercises with youth that are just coming in, these guys are leading the way and catching everybody up on our progress and what it is that we've learned so far. Hey, Agnes, are you on the line?
I'm here.
Nice. Let's tell them a little bit about our project and introduce them to our Instagram, if you don't mind.
So we recently started an Instagram where you guys can follow us. It's called Mass Remediation, and it's starting off, so it's still new, and we mostly post stories, but we have a few actual posts. And if you follow us, you can get updates on our journey learning about bioremediation and our work that we do and our updates. So you can get updates when we get them, when we go out to go see the area. We also have for the work that we do there at Franklin and Tapley, and the work that we learn when we go visit other places too. So feel free to follow us. We have 24 followers as of now. Let's get us to 75 by the end of August. So yeah, give us a follow.
Nice. Yeah, man, it's good to share our story. With this project, we also want to take some histories in account for our neighbors. What have our neighbors seen in our sites? What have they experienced thus far? How do they feel about the work we're doing? So in that process as well is our own histories. We're going to be the elders one day, and it's important we tell our stories. So the documentation we're doing, we want to figure out different creative ways to share it with you guys. And, we want everybody to be a part of this process. The neighborhood comes by and asks questions, and a lot of times it might be something that I don't might not know, but the important thing here is we understand how to find it out, and who is our project partners or our allies, how to do the research. It's been very great experience to document and learn about, like, a lot of these things. I bet some of you guys will recognize in your own yards. We got some blackberries, some raspberries that we planted on site. And we're just understanding the different depths, the root structures, and the different space that all of these plants occupy. And if you see this bare soil, so we learned that we're going to cover any of this bare soil with materials such as grass clippings and shredded leaves and things of that nature, some mulch, hay, straw, so we can keep our soil covered and healthy. As you see, we have different forms of life here. And as she mentioned earlier, we had a furry friend who seemed to find a snuggly space in there. So, we're not sure exactly what that means for our site, but it looks like we're creating an environment for bacteria to fungi to bugs all the way to mammals. I think it's just interesting to know. This is Sister Anna, working hard and great with us. She's the one that is the glue and the binding, and we have to pay special attention to the people around us that are there for us all the way, and just acknowledge them in any way we can. You see our site, we still have some bare spots that we're looking to cover, but we're learning a lot. We're doing carbon proxy testing, and we're analyzing the soil in different ways. So we're digging in deep, and when we're getting in there, we're talking about what we're seeing. We're studying the outside, the control, and we're studying inside as well. We're looking at different things like root space, how much roots are occupying the soil, how deep is the furthest root that we can see, how many earthworms are there, what is the texture and the color of the soil, and how does it smell and feel in your hands. So we can really understand our soils and try to take notice of the differences as our work proceeds. Here we are looking at aggregates and soil root structure and how well our soil's doing. And sometimes when we would do a slate test and we'll put an aggregate in there or a clump of soil, and we get a big cloudy mess like this, and that just goes to show you how poor the soil quality is. It's not holding together. And so before we can think about trying to deal with the lead and the arsenic or whatever levels that we're finding that are elevated, we have to make the soil a healthy place for the microbes and bacteria to function, and for the roots of the plants to take hold. This sample's a little better. This was inside our plot, just after a year of adding compost and plants. Then we do the texture by field procedures. So we're like rolling it out in ribbons, and just to see how well it stays intact, if we have a clay soil or sandy soil. And we're taking note of it all along the way. This is our infiltration test. We're setting up a ring, and then we're filling the ring with water, setting a timer. And this will go ahead and tell us how long it's going to take for that water to infiltrate the soil, and tell us a little something about how well the soil will retain moisture, or how well the water infiltrates the top layer of soil. So yeah, this is Bernard again, and Josh, and Sister Anna, and these guys, if you see them in the neighborhood, give them some respect, because these are our soil scientists. Like, they're doing these experiments that might seem basic, but gives us some really qualitative work and observations. This is how we take some of our notes. To see, for example, we look at what we see, whether it's a cat or the soil conditions, or how much of our compost gone down. Is it time to flip it? And we take notice of our slate test, our ribbon test, water infiltration test, and our pedometer test. So that way we'll measure compaction as well, and all the different things. So yeah, this is our fungi that we're growing, and I was wondering if Nancy, if you can speak on some of this. some of these pictures, like this picture here, is from our compost pile, and then we have some others that will go along the show. But we're just wondering, because I know we did some inoculations with bacteria from products from AEA, Advanced Eco Agriculture, and then we did inoculate some of our straw and wood chips with wine caps and oysters. So I was wondering if you'd be able to speak a little bit about what we're seeing here, especially after all this rain in New England.
Well, I'm not going to be able to identify all those mushrooms, and you have quite a few of them on this, but I do see a couple that I can identify, and I can talk a little bit more about how to identify and why we can get excited that mushrooms showed up on the site we didn't necessarily inoculate for. So two of the mushrooms I saw in this presentation were Caprinus comatus, which is shaggy mane, and then here we're looking actually at Coprinopsis, which is inky cap, and both of these mushrooms are remediators, and the fact that they just showed up in this substrate that you thought you had oysters in, or you had wine caps in, it's fine. You might not have the wine caps and the oysters, you might have these other mushrooms, and it's really great to study that and to document who's there. So one of the ways that you can make fungal observations is you start looking at parts of mushrooms, and so you could start taking and there's some slime mold on the right there. But what you could start doing is start taking some measurements and doing some spore prints and collections, and then look at mushroomexpert.com, which is an online key tool where you can key out your mushrooms to identify them. So you can start learning about mushroom parts and understanding who's there, because if they're happily there, just like there's a nice one, a stinkhorn. So you can see the fly being interested in it because it's so stinky. But what would be great to do is realize that just like all the weeds that we saw earlier, where what we consider weeds are, those plants are doing the work in the soil. And if they're really happy someplace, they're probably remediating the soil. So a lot of things I saw are known remediators. Here's some coral mushrooms. So I would start learning some mushrooms and see who's showing up in your mulches and in the compost bin, and start learning how to key them out to find out if they are known remediators, or if you can start introducing them to the remediation site to test them to see if they will help in remediation.
I think that's a beautiful thought, that these things that are showing up, whether it's weeds or some kind of fungi, can actually play an interesting role in the remediation process and be the actual, maybe the winner of it all, you know?
Yeah, definitely.
We're learning about mycelium here, and I'm kind of like infatuated with it. I see a mushroom and I'm like, man, I should leave that alone. But I just want to like look under it and just see where it's coming from, and understand that it's really just the fruiting body of something much bigger underneath. I mean, look at this guy here. So yeah, we want to see our soils take hold, and like I said, this is important in many ways. If we can take the little spaces that we do have in the city and make them viable for production and growth, and benefits are insurmountable. So Nancy, what we're looking at here is our second test site and it has only the cover crop in it. And we were talking a little bit about the differences of what this site might represent and tell us compared to a site that it's full with different flowers and stuff. Can you maybe speak to that a little bit?
Sure. So what's interesting about this, the fact that you have two sites, is one, you have planted with a diverse amount of species, and the other one you have planted in a monocrop. I see some other plants in there, but it's mostly a monocrop. And so you have two distinct lines of investigation. One is, how does this one plant kind of affect biology and also remediate soil forward? And then you have the other plot that you have so many different plants that you can test for the same thing. So I think it's a really great comparison, because most people, when they're doing remediation, tend to plant one plant. They're not planting a diversity, but we wanted the garden to have a diverse representation of plants for the pollinators, and for to have the community of plants being more diverse. Here's two different vetches, which are great, legumes. Also, both of them known as remediators. So I think what's going to be nice is that you have two different sites functioning, one in a very narrow way or very focused way, and the other one in a much more kind of complex way. And you'll be able to look at how they work with the soil differently over time.
Nice. Yeah, so looking towards our interviews of those in the city who live and work in the spaces around us and what this project means to them, look for our Instagram page to find out, what it is that we're missing in this process, which is the way people feel and think and what they know about our spaces. Also, if you have any links, book recommendations, places to visit, or anything that we can learn more from, we're willing to soak it all in. We know there's a ton of stuff out there, and we're just on the tip of an iceberg of learning what we can here. And yeah, so do we have any questions for Nancy or for Agnes?
Wow, what a wonderful presentation, and pretty in depth into the second year of this wonderful project that I get the chance to witness every week. So at this point, folks can feel free to either place their questions in the chat. You can put your questions right in there, and Christine and I will pull those questions. Christine is my co-pilot for today. Also, if you would like, now we also want to take advantage of our Zoom norms. Everybody's been on Zoom by now. So if you would like to ask your question out loud, we welcome that. Just take yourself off of mute, ask your question, place yourself back on mute so we maintain the sound quality for everyone. So at this time, please feel free to put your questions in. I'm going to start off, as they say, get this party started. Oh, great. From Brownfield sites. I'm so glad that question came up before I threw my question in the ring. There's a question here concerning where were the lots capped as brownfield sites? That's a great question. So that's open up to the floor. Thank you for that.
That's a good question. Were they capped as brownfield sites? I don't even know what that means. If honestly, could you explain maybe what that means? I'm imagining. I do think it was a matter of fact. Beth, are you on this call right here? Yeah. Just if you can let us know what you mean by brownfield sites. I think I understand that it's been covered with topsoil.
Yeah, I was. Can you hear me?
Yep.
It was the difference between just vacant lots and maybe lots that were used to have a factory on them. And as the factory was destroyed, then it left the contamination in the soil. And then sometimes they would just put a cap on top of the on top of it to contain the contaminants. And sometimes, like projects like this, they're worried that the roots will go through the cap and then release the chemicals that might be underneath. So maybe this area was just vacant lots from a housing place, I would imagine, or.
Yeah, I believe that there were two buildings on this lot. I don't know if Sister Anna or Beth, if you guys have exact answer to that.
And Beth Ward, who's joining us, she is my counterpart at Home City Housing. She works predominantly with the youth in housing development. My understanding based on the map, and Beth, who is a native Springfield resident, she probably has more of the history. But
Okay.
understanding that there was a house with a garage. When we took our first soil sample, it did come back with some contaminants. We could tell that there was some kind of garage there. So Ben, to your knowledge in this area, was there a structure or several structures at the time? And Beth, you can just take yourself off of mute to answer that question. Okay, she may be having trouble off, but that was the history that I have.
Yeah, it looks more like in a residential area.
Mm-hmm.
Garage, okay.
Yeah, we remember seeing the driveway and then the garage, and that was the last picture that we seen with a building on it.
Oh, okay.
Mm-hmm.
And our consultant Leah had told us that there was lead levels. There was very high lead levels in all of the samples. Arsenic levels were elevated, and the carbon ranges that were found were low. And the issue that comes into play to consider is that these can be uptaken by plants, but can also be breathed in and handled. So, we were thinking about different ways to approach this project and, think of the safety, but then also taking a step back and realizing that it's literally like that in every lot that we're dealing with in Springfield. And so when we're learning about these contaminants and like how we should be around them with gloves and rubber boots and stuff, it's like, wow, like these are the lands that are our playgrounds and our our community spaces. There's something to think about there.
Mm-hmm. Yes, excellent question. Thank you for that. Myra has a question concerning what do you mean to key out mushrooms? So Andrew or Nancy, I don't know if you want to take that question.
Yeah, I'll take that one. I mean, so Myra, I don't know if you've seen plant keys or and they're basically just kind of, it's like a tree of information. So you kind of look at what you're looking at and you go, and it'll ask you a question. It's like, did you find it on it? For example, with a mushroom, it might say, did you find it on the trunk of a tree or at the base of a tree? Or maybe you found it in grass, like where did you find it? And then it tells you if you found it here, then you're gonna start pursuing this direction. If you find it over here, you can pursue this direction. And it basically, through various questions, gets you to the point where you get to the mushroom that you're looking at. It does take you learning a little bit about mushroom parts and what they are, from cap to gills or pores and teeth, or the stipe, the color of the spore print, where it's found, measurements of all those things. So it can sound really intimidating, but it just helps channel you into towards what you're looking at as you answer the questions of what you're seeing. And the same thing exists for plants or trees. And mushroomexpert.com has a really extensive online one. And like, he'll start with like, what tree was it found? Was it found by a pine tree or a maple or something like that? Because most mushrooms have a association with certain trees. They're found in certain places. So it's not just the mushroom itself, but where it sprung from. and what time of year it was, etc. So that's, if that answers your question. So where do I find a key? So again, mushroomexpert.com has one online, but if you get a just Eastern mushrooms of North America, or you find another kind of really basic guide, it'll talk about those mushroom parts that the questions are going to ask you about. And then there'll be some keying out that are specific to the Northeast for mushrooms that you see. And yeah, so most books will have them. And then I like mushroomexpert.com just because it's really extensive. Yeah, so hopefully that's helpful.
Well, that was a great question and follow-up question from Myra, and a great response. I'm going to pose a question really for both Agnes and Andrew. As this project proceeds, we're in the middle of our second year. What are some of the things that you're hoping to see as we move into the end of the third year, which is 2022? What are some of the things, Andrew and Agnes, that you're looking to learn and gain from this project? And either one can start first.
Yeah, I'm looking to learn all I can. I think an important part of this is plant ID, mushroom ID. So being getting familiar with what it is that I'm seeing. A lot of times I might think and try to jump ahead to like some kind of secret recipe or formula. And, what's the science project to do here next? But I think really if we could just identify and know what it is that we have already in front of us, will be a big key. All the way down to like some micropsy. Am I saying that right? Where we can see, what the eye can't see with some tools and microscopes. So and then I would like to see the sites more heavily planted, and some of the results of the tissue samples that we are going to send in to see, what it is that we're actually eating and putting in our mouths. Because a lot of times people are excited to grow and they'll just grow in their backyard. And we don't want to scare people away from doing that, but educate them in a way to do it safely, and in a way that we can understand as well.
Excellent, excellent. And Agnes, as one of the key youth leaders, what is something that you're hoping to gain from this project as we move into the third year?
Well, this was my first time working with mushrooms. I knew nothing about it at all, except what I would learn in like biology class. So I'm hoping to learn more about and have a better understanding about how mushrooms help out the ecosystem with the soil. So it's really nice seeing it hands-on, having like a one-on-one experience with it, and because I work better hands-on. So I'm hoping to learn more about that and more about how to learn about soil testing. So this was all really new, so I enjoyed that. I'm hoping to see, what I'm hoping to see is more animal life being brought in there, and more community involvement, since we are starting to see that, which is something really cool. And I know we have the plants that we planted there, we aren't able to eat at the moment, but we do have a compost bin, and I'm hoping to now see the plants grow and us being able to add it back into the compost and add it back into the soil. So I'm really excited to look forward to that.
Excellent. And Myra has another great question. And thank you, Myra, for giving me some context of where North Andover is. I'm still Massachusetts geographically challenged at times. But she asked, is your project exclusive to Springfield only? Can you set us up in our community? And she is in the North Andover area near Lawrence. And Andrew, any thoughts about moving this out of Springfield once we get some results back?
Yeah, like you said, there's many cities. This is not a unique case where we're dealing with potential contaminated soil. And I would like to see the youth Agnes and Bernard and everybody else to lead these type of endeavors. And it's just a matter of how do we make those connections and communications? I think it's important that we do that, just to cut down even on, you know, our involvement in the entire food system. You know, we can cut it down, you know, and like neighbor to neighbor, we can learn from each other. So I mean, it all sounds dreamy to me, because sometimes it's like just the how, you know, like if you're in North Andover, let's make it happen, but just how.
Mhm. And Myra, I can add, since as a food access coordinator and a member of the soil health technical assistance team, we're on here with Christine Manick, who is a soil health grant writer. We can look into how we can get funding for your area. As Andrew mentioned, this is something that is not unique to Springfield. It is all over. We moved here from Baltimore, Baltimore, Maryland, before coming here, and they can't go in ground. They have to do mostly raised. Jersey City, same thing. They cannot go in ground. Boston, very few places, just because of this one issue. So definitely we can look into how we can partner with you, and we'll put our contact information towards the end. Good question, and it's always great to make a connect. So thank you so much, Myra, for that. I want to raise a question to both Nancy and Andrew as it relates to soil testing, because we know testing for this kind of thing is not just related to the soil, but we can also look at what the plants are taking in. So I'm going to start with Nancy first on this question. How does testing tissue of plants, leaves and so forth, what are some of the things that, I don't want you to put a prediction out there, but what you would like to use as a tool to kind of help guide the remainder of this?
You cut out a little bit. So your question is about what can plant tissue testing help?
Yes, what can plant tissue show us?
Well, plants are situated and in one place, and so and they have to get everything where they're at to be healthy. And so they're working a lot of relationships through their roots, as much as they're working relationships through their crowns. And the relationship with the roots are with bacteria and fungi and other microorganisms. There's a lot going on in that rhizosphere, that root zone. But what they do is they send out sugars to attract those microorganisms, and the microorganisms helps them bring minerals in a usable form into their bodies. So as they reach into the soil, and having all these chemical and biological reactions, they're also bringing things into their bodies and sequestering them. So as they build their bodies. So at the end of a season, plants will show growing. They'll show some of the nutrients in the soil where they're growing. And so you can cut some of those plants at the end of a long growing season and test their tissues for heavy metals, for their nutritional content, etc. So people are interested in testing tissue just to have, better tasting vegetables, and do they have the nutrients that you want in them. But we can also test them for metals. So again, a lot of the bacteria and the fungi in the soil are doing a lot of that. They're binding things, they're decomposing things, but the plants usually take it on and hold it within their structure. So they're really good short-term testing to do, because it takes a while to remediate soil. And in most remediation projects, we only test the soil every 3 to 5 years, but we test the plant tissues because they're there working every year. And we can find out what they're able to take out of the soil and what relationships they have. So that's something that we're really curious about, what tissue tests are going to yield, tell us.
Excellent, excellent. Christine, I know, has some questions that she'd like to pose to you as well. So Christine, go ahead, take it away.
Thanks, Anna. One thing I was wondering was just kind of, Andrew, if you could reflect on some of the successes and challenges that you've had with the project and how you might work upon them moving forward along the lines of what Anna was saying and what you're hoping for in 2022 moving forward.
Yeah, I'll start with the challenges just being, you know, there was some switches as far as our site as first, for different reasons, which site we were working on. And then, you know, the contacts for those sites. And so just having somebody on site who is consistently on the project has been a little bit of a challenge. And then, well, also, you know, just seeing the growth of like the evolving of the project. So in the grant, we wrote in a lot of money for testing, soil testing, which now we learn, as Nancy just said, we don't necessarily have to test every couple months, every few months, where, you know, we can test a little less in the soil and do some of the plant testing. So money is moved from, you know, here to there. And on the other note, like what I've really liked that I've seen as a success is just the youth that have been involved, their leadership and their pride and standing in the site and looking around them and realizing the work they're doing and the importance of the work they're doing and owning it, you know, and owning the spaces we're in. I think that is the most important thing of it all. And yeah, so I think that's been a huge success for this project. And a lot of the things going into this project that I thought we discovered, like really scientifically, I realized that there was a lot more to remediating soils, particularly in the city, than just taking a sample and realizing it's contaminated. There's a lot of levels to this, and I was exposing a lot of the levels, and you know, I hope that people can, you know, learn from this from all angles.
Thank you. That's really interesting. Is the community's response fairly positive as well, kind of mirroring that of the youth?
Absolutely, yeah. And the people are walking by and asking questions, and just really giving a shout out to the fact that we're doing the work. On Franklin Street, there, you know, it's a lot of questions of because of the way the plot's situated, it's kind of like in the middle of a bigger area, so there's a lot of curiosity. Whereas on at GTC on Hancock Street and Walnut Street, we're actually making a lot of changes, and a lot of those changes are doing it intentionally, knowing that we have to take care of our soils and make them viable for production. A lot of the area, most of the area at GTC has been tested over the years, and whether they knew it or not, they were doing remediation there by building their soils up. And there's like a particular lot in the back of one of the lots that we're focusing on, as we knew there was some possible hydrocarbons and leads in that area.
Thanks. Agnes mentioned that in Franklin, people are spending more time near the area this year. Even the kids are playing that near it now too. That's it seems like just kind of a fantastic secondary result of it as well. I'm sure it's hopefully creating a little bit more of a community space, I imagine.
Yeah, and then we got to think about it too. It's like we might go into these spaces like, oh, I'm going to build a community garden here and they're going to love it. But we have to find out, what does the community want? You know, they may not want to see that. There might be people that hate bugs and don't want to have bees flying around their heads, or, you know, they want to rather have a pool or a park there. But to understand that and to hear their wishes, and then later down the line say, man, I can really see a community garden here, or I understand what the work is that you're doing. Yeah, I still want a pool, but I really get it. You know what I mean? So I think there's a lot of beautiful interactions. You know, we want to farm. We want to look at, like, how do we produce food for our community, right? Well, there's a lot of things that we want to look at, and like, how do we get the most production out of this zucchini, and how do we trellis our tomatoes? But we can't even get there until we figure out the science of remediating our soils, because they're potentially contaminated. So there's like a real a thing there. So if you see an urban grower, you know, give him a heads up, because he's doing some work that is needed in that community. A lot of our food is coming from outside of our community, and I don't think that, I think it's very hard to feed a population without doing that. But I think there's some power in being able to grow your own food. So this is like this project is a basis for a lot of development in many ways.
Mhm. And if I can add too, this is a kind of a partner project to the Tapley Court Apartments Community Garden, which is located in the Bay Area of Springfield, which is a part of Mason Square. Both areas do not have a large supermarket like say a Shaw's or a shop nearby. Mostly smaller convenience stores, bodegas, a lot of fast foods, lot of grinder shops. Now you're from Rhode Island, you may not say grinder, I don't know what they call it. But it's nice to be able to do this kind of work in those areas. And Andrew brings up an important part. That's important that we're able to grow food and to grow safely. Christine brought up a thought, if I can interject this question concerning the oral history, because we're going to document our results, but we're not going to do the typical academic white paper, correct? So the oral histories, Andrew, you want to, and also Nancy, I see you do a lot of oral history in the works that you've done in other places in the country. Can you both speak about the importance of the oral history and what it brings to a project like that?
You first, Andrew.
Yeah, I think I think the oral histories are very important, and I think we're ready to go ahead and start asking some questions to people that we wanted to know, but we realized the magnitude of what we're going to experience, and we wanted to properly document it. So we're thinking of ways of different creative ways, even if it were a comic book or a story with more visual, using some type of document online. But in any case, we wanted to properly document what we're going to hear. We've had experiences with some other projects locally where, for example, Black Holyoke by Erica Slocum, she is doing work that is recording people that were in Holyoke that, a lot of the Black history in Holyoke you don't think about, when you're familiar with the population that exists, you don't realize that, there's much more to the story. And that's another reason why telling our story is important, because if you don't tell it, somebody will tell it for you. And so I know Nancy has done a lot of powerful work with this too, and she gives us some very good things to think about when we go to do our interviews. Nancy would like to add to that.
I think I'll just add is like, a lot of stories haven't been recorded and they'll be lost and we won't even know they're lost. And so the idea is to create a space for those stories to come forward, to understand place and how people identify with that place and how they care for that place and histories that they have with their own experience of place through their families or other people they associate with. So it's about really getting to having those stories come forth so you can hear how place is enlivened and engaged, but also really understanding place in a more complex way. I think so much of our work is very this is what we need to do, this is how we do it, and it's really narrow, and each of our places have so much uniqueness and character and histories and traumas. So I think it's really important to open that up, and when you start talking to people and what they remember and what they've experienced, you learn a lot about place. And so that's a really important piece, and I see that as part of remediation as well.
Okay. And there's a question here in the chat. Have you thought about how Instagram links to oral history? And Agnes, you can jump in too as the creator of our Instagram page. But what are your thoughts about how linking not just Instagram, but Facebook, TikTok, all that to oral history? How can that work in a project like this?
Me or Andrew?
Well, Agnes, why don't you go ahead and get us started?
So when we were making the Instagram page, we originally was doing it while we were part of a Zing project for oral history, and we were trying to come up with ideas and ways to get the community involved and stuff. So we had that in mind when we were making the Instagram page, and we did mention about how ideas about how we can include the recordings maybe in it. So it's still running, it's still starting up, and we're still getting the hang of it. But yeah, the whole goal for the social media, I believe, was to have for other people to learn what we're doing and to get the community maybe involved. We did mention about how maybe we wanted to right, Andrew, how we want to maybe put the link somewhere so that the community, when they pass by the areas, they can see it and get all the information.
Yeah, absolutely. And then they can leave links and pictures as well from their view and their perspective. And you know, we're in the age where a lot of people are on social media, and a lot of the news and reports and what people are learning is on these social medias. They're waking up and they're scrolling. So if we can, you know, spread our knowledge through that, then so be it. We should do it. You know, and we're talking about community. So when we think about advertising and spreading knowledge, you know, maybe we can look at newspapers and think about, you know, what demographic do we want to see receiving this information, and get them that information.
And Nancy, in the oral history work that you've done in your projects, does social media play a part?
Oh, not so much for me. I let other people do that. So right now I have a urban field assistant named Valentina, and she is all over that. That'll come out more. And so I was listening to you and I thought, what a great idea. So Agnes, that's awesome that you're really holding that and pushing that forward. I think I wouldn't have thought about it just because of my generation, you know. so I think that's great. So that'll happen, but not through me personally.
So, wow, as we begin to wind down, this has just been such a wonderful workshop in terms of looking at this type of work in our backyards. And I'm excited to see the results, because I can see where this can go as we look at a lot of our urban centers and the struggles they have with growing in contaminated soils. So as we are beginning to come to the 5:30 hour, I'd like to give this time for any closing comments from Agnes and Andrew and Nancy, and then we have some announcements. So let's start with Agnes and then go to Andrew, and then Nancy, you can, as I say in the church, take us home and give some closing comments about the project so far.
I just want to say I really enjoy being part of this project, and I can't wait to see what the area is going to look like even in the future when maybe none of us are on. I imagine what will the area look like in like 5 years. So I'm excited with that. But I do want to say, again, you guys should all follow us on Mass Remediation, both sites of Franklin and the Tapley site that was mentioned too. They're both featured on Mass Remediation. So give us a follow on Instagram. Thank you.
And Agnes, if you can do us a solid and put that link in for Mass Remediation so that folks can like that page, okay? Andrew, any closing comments from you?
Yeah, we're excited. There's a lot of things that I couldn't catch in my slideshow. I had 100 photo limit, so I wanted to really express what it is that we did. And we're going to be looking at ferments. So we got a comfrey ferment that we've been working with, and we're going to do some nettles ferments, and we'll be learning a lot more about that and what that means and what we'll be introducing into our soils. So we would like to stay on with learning and just keep it coming. I want to have more conversations with Nancy, because I get a boatload of gold every time I do. And I think that's what's important, is those connections between folks who have done the work, who understand the work, and then those who can be representative of that work in their communities. I think it means a lot, I would say, to see Sister Anna and myself in these communities working with people who look like us doing the work. So I think that goes miles. And if you guys are looking for fresh produce in Springfield, come by GTC Farm Stand, 200 Walnut Street, and visit our website, because we as well want to, like, you know, provide workshops and things like that, youth-led workshops, so they can show you the work that we're doing. And we have a curriculum now at GTC, so we're on track to providing our youth with the tools they need to become the leaders that we would like to see.
Mhm. Excellent. Thank you. And Nancy, like I said, you can take us home.
Well, let's just say I'm in Chicago right now, and I have spent 2 weeks looking for clean topsoil. And I'm telling you, I need 2 semi trucks worth for this project, and it's really hard to find. So we are in the moment where we really need to do right with what we have and get it better, because we're not always just going to be able to find some clean topsoil, which is usually taken from a meadow or some other natural system. To service us. So I think this whole idea of bioremediation is reclaiming the land that we've neglected or abused before. And I'm really excited that you're all doing this really powerful work, and excited by it.
Wow. And
David.
it is indeed powerful. You hit the nail right on the head, because it, you know, for folks like Nancy and myself, when we see our youth and folks like Andrew engaged, we know our future is secure. So I think she hit it right on the head. I'd like to take this opportunity to thank Nancy Clem, our consultant on this project. She has been a wonderful help and a source of information. Big thank you to Andrew, my colleague and brother in this. It's wonderful to work side by side with him, with a wonderful group of youth in both sites. And then finally to Agnes, leading the youth out. She's also NOFA Mass's intern. She interns for the Food Access Program. So I get a chance to work with her day in, day out, and she has been wonderful. And I also want to give a shout out to Beth Ward. She is the Resident Outreach Coordinator for Home City Housing. She is the one that helps me get the youth together, helps with the field trips and so forth. We could not do any of these projects without her. And so as we begin to wind down, Christine is going to put a few links in the chat that I'm going to refer to. I'm going to share my screen real quick, because I think I finally got the thing to work. First of all, we want to thank everyone for your patience with us as we work through some technical situations. We know how technology is. It's not always kind to us. So we really appreciate your patience with us on that. I just want to show you a few things that we couldn't get to. So Christine will put these links in. One, we have an evaluation that we want people to check out, and she'll place that link in the chat. We also would love for you to go visit our vendors. And there's a link for that. The vendors also include some of our partner organizations that have helped sponsor this. So she'll also place that link in the chat, as well as where you can find the indigenous families and First Nation peoples that are in the areas that we now inhabited. Because as I mentioned before, there are First Nation, First World peoples who were here, are still here, and continue to steward this land. Also, we have a wonderful auction going on. Please check out our auction. You'll see the link for that in the chat as well. And lastly, please continue to visit the other workshops. We have a whole bioremediation track. Willie Crosby will be talking about the role of mushrooms, and we use some of his work in this project. You did hear from Debbie Cook concerning biochar, and then we'll hear more about indigenous microorganisms. You saw the IMO traps. I'm the one that wanted that pink color in the indigenous microbe traps. I kept saying, oh, where's the pink? I want the hot pink. And so we have Marco that will be talking about that in another workshop. This is all part of the bioremediation track. And then my colleague here, Christine, will be hosting a workshop as it relates to soil health. And Christine, I believe that's in reference to the carrots and spinach project that we've been working on, correct?
We're having the soil health tech team discussion on Tuesday afternoon, or Tuesday evening, if that's the one you were thinking of.
Yes. Yes, that one, I'll be on that panel. So a lot more to come in this conference. Thank you for joining us. Any questions, I'll ask Andrew and Nancy to put their contact info in the chat. And then you can reach me at anna@nofamass.org if you have any other questions. We so appreciate you. Please come back later on tonight. We have more of the 50th anniversary of NOFA. So we have a special event tonight that you don't want to miss. We had a great one to kick off the conference last night. So please join us as we are also celebrating 50 years of NOFA Mass. So everyone, thank you so much for attending. Thank you for spending this beautiful day with us. Look forward to seeing you during the rest of the conference. Be well and enjoy yourself. Thank you so much. Good night.